everyone. My name is Dr. A. Rajni, Head Department of Botany and Food and Nutrition, RBVRR Women's College, Narayanguda. Today we will discuss about the evolution of sporophyte in bryophytes. What is the evolution? How to evolution to starts in bryophytes? Mostly bryophytes is the dominate for the gametophytic stage. But what is the evolution? How to evolution to start in the bryophytes? These are things we will discuss about to the bryophytic plants. First we will discuss to the what is sporaginum. It is sporophyte of bryophytes. It consists of a single terminal sporangium with a bulbous fruit is there, and which are with or without an unbranched stalk or seta is present. The sporogonium is a very delicate, short-lived and nutritionally dependent on its gametophytic stage. This is the sporophyte. What is in this sporophytic? There are the controversies or contrasting views are there. Two contrasting. One is the evolution of sporophyte by progressive sterilization of potential sporogenous tissue. This is called, the theory name is called progressive evolution sporophytic theory. Coming to the second one, evolution of sporophyte due to progressive reduction or simplification of called, that is the theory of regressive evolution. These two theories are very important for evolution of sporophytes. Coming to what is the introduction of sporophyte? The sporophyte of bryophytes is a diploid multicellular structure that developed by the mitotic division of a diploid zygote. Here zygote meaning oosphere. In this cell within the archegonium born on the gametophytic 4. It produced haploid spores after the meiotic division of sporogenous cells that are differentiated within it. The sporophytes of bryophytes shows a very great variety of tissues differentiation from the simple to complex forms. There are two main constructing views are there that have been suggested to explain to the evolution of sporophyte. Two types of theories are there. The theory of progressive sterilization of sporogenous tissue, and coming to the second one, the theory of regressive evolution. Coming to the first one, the theory of progressive sterilization. This theory view also known as the progressive evolution theory sterilization. It was originally proposed the scientist BRAOU scientist in 1908 and 1935 and supported by the another scientist Campbell the year 1918 and 1940, and the Cavers, another scientist Cavers in 1910. These are scientists to explain this theory. They assumed that the simplest and most primitive sporophyte from Rixia, the higher and more complex type of sporophyte, for example Funaria, Sphagnum, Pogentum, these are the examples, have evolved by a progressive sterilization of potential sporogenous tissues. These progressive evolutions are according to this theory. Complex form Funaria, Sphagnum, example this one. Another one potential fertile is the Rixia and Marchantia. This theory approximately 50% of sporogenous cells form spores and the remaining 50% of cells form sterile illators with characteristic spiral thickenings. Thus with the bulbous fruit and elongated seta and a single layered capsule wall and large number of illators are formed as a sterile continuous the sporophyte. As a result of this complexity, a mechanism spores dispersal has been established. The further sterilization of sporophytic tissue can be seen in gingermanials. The example Pelea. Here the sporophyte shows differentiated in foot, seta and capsule but the jacket of capsule is 2 or more layer structures are there. Furthermore, a fixed sterile elatophores is present at the proximal end of the capsule with a bunch of elaters. The elatophores may be at the base as in Pilia or at the apex as in Rixia. The actual sporogenous tissue has been reduced to the comparatively small percentage, that is only for 10% of the total sporophyte. Coming to the mature sporophyte of Anthoceras, consists of a bulbous, fulbous foot and a horn or basal part like capsule like are there. The capsule grows continuously due to the activity of intercalary meristems. The entire endothecium form the sterile columella. The amphithecium forms capsule, capsule wall and sporogenous tissues. The wall of capsule is multi-layered and it surrounded by a narrow band or present in the sporogenous tissue. The increase of sterilization, the sporogenous tissue from simple sporophyte of Rixia to the most complex type of funaria can be arranged through the different types. The simple of sporophyte bryophyte is the Rixia. In all species of Rixia, the sporophyte consists of only capsule, there being no trace of the seta and foot. The second stage, this is the second stage seen in Corsinia belonging to Apatecapsida, where a very small sterile foot develops. The capsule has a single layered jacket, but inside the capsule some of sporogenous cell instead of forming spores from sterile nutritive cells. This is the second stage. Coming to the third stage, further sterilization is seen in Sporocarpus, where the sporophyte has a sterile bulbous foot and a narrow seta in addition to fertile capsule. See in the diagram here, the mature sporophyte in the Spirocarpus. Here you can observe the foot part is very reduced. This part is the foot, this part is called seta. Here you can observe this is the sterile cells jacket layer. This one is called the mature sporophyte jacket layer is there. Inside sterile cells are present. This one is this picture shows that one mature sporophyte. And coming to the fourth stage, this is the Corsinia. Here this stage is represented by Targonia, where the sporophyte consists of a sterile bulbous foot. Here you can observe the foot is very bulb like structures, reduce and bulb. A sterile narrow seat and fertile capsules also present in this one. Thus in Targonia, more sterilization of sporogenous tissue has been observed. You can observe the picture of the Corsinia. This is the mature sporophytic in Corsinia. You can observe in this sporophyte, here the bulb, this is the foot part, this part is called the foot. In this stage here consists sterile bulbous foot. What is the foot name this one? Here sterile bulbous foot is present and sterile narrow, you can observe the way this part, sterile narrow seta and present. And this one is the fertile capsules. These all inside fertile capsules are present. This is the fourth stage example for the Corsinia. Coming to the fifth stage. The fifth stage, this stage is illustrated by the Markantia, where further sterilization of sporogenous tissue has been noted in comparison with Targonia. In Markantia, the sterile tissue consists of a broad foot-like structure, a massive seta, a single layer of jacket of capsule. Here, sterile apical cap at the apex of capsule and a large number of long elyters with spiral thickenings are present. Come see in this picture, it shows that one evolution of sporophyte in bryophyte. The first picture can observe, that is the Rixia species. Here Rixia species, this is the layers, this part is the food part, and here in this one jacket layers are present, and inside the spore capsules are present. Coming to the second one, Spirocarpus. The second B picture is called the Spirocarpus. This Spirocarpus, one is the food part, bulge part is the food, and this part is called the seta, and here in this one capsules are present. Inside capsules, here you can, this is a jacket like structure. In the jacket inside you can observe that one elyters are present. These are called, all are the elyters. This one is the Spirocarpus picture. Coming to the this part, this one is the Targonia. In the Targonia also, this is a foot and seta, sterile foot and sterile seta, sterile is there. This foot also very small in size. This one, another one is a seta, also very small, and very few ranges are present in the seed part. Coming to the this part is the jacket and capsule, the very large size is present in the Targonia. And coming to the D, Hypophilus. These hypophilus, the food part you can observe here, very important. Hypophilus, the food part is the well developed, to starting the development point to the foods are present. Many tissues also present in the food region. And another above part of food, this one is a sterile part. The total is called the sterile part, and this is the capsule. Inside the capsule is the elyters. See, many elyters are to present inside the jacket. Coming to the E, Pilia ephilica. In this one Pilia ephilica, and coming to the difference in between Markantia and Pilia. This is the in the Markantia, the development of sporophyte, the food style and capsule is the enlarged to compare other three pictures. And here Pilia epophyte also, this part bulge of food part is there, and this one is a seta, and here jacket or capsules are present. Inside the elyters also present in this one. Coming to the sixth stage of sporophytic tissue. This stage is represented by some members of Gingermaniales class. This like example Pilia, Ricardia, these etc. are in this stage. Here more sterilization of sporogenous tissue has been observed in this one. This sporophyte is differentiated into the foot, seta and capsule, having the multi-layered jacket cells are present. The sporogenous tissues produced mass of sterile elytophores and diffused elyters also present. Coming to the seventh stage of sporophyte. This stage is illustrated by members of Anthoceratopsida or Anthrotophyta, like the best example for Anthoceras. Here marked reduction is in the sporogenous tissue has been noted in this Anthoceras. The multi-layered of capsule differentiated into the epidermis with stomata and chlorophyllous cells also present in this seventh stage of Anthoceras. Coming to the eighth stage, this one is the final stage. The total members of Bryopsida like Funaria, Polytrichum, Pogonatum, etc. shows the highest degree of sterilization. The sporophyte is differentiated into the foot, a long sheet and a capsule. The sterile tissue of capsule consists of a apophysis, operculum, many layered jacket cells, the columella, trabeculate, the wall of spores sac and peristomes also present. This point is very most important for the peristomes. The sporogenous tissue is restricted to the spore sacs only. This one is a enlarge of the sporophytic anthoceras. See here food part is the enlarged and this part is the capsule, columella and capsule. See here in between capsule wall and food part, the sterile where the sterile part is there. In this one foot and capsule, both are the sterile tissues are present in this part, and where the seta is there in between the foot, the foot and seta and capsule, meristematic zone is present. Very important characteristic features are present in the anthoceras to start the meristematic tissue in this one anthoceras. This is a very important character for the anthoceras. Coming to the funaria. This one is the funaria mature capsule. This here very big capsule are there. This part is the foot, seta and this is the columella or caps. And this is a funaria plant. In this funaria plant, most mosses, funaria, sporogenous tissues are also present in this one. Thus way the rhexia of the thus the sporophyte of rhexia is the simplest among the bryophytes with a very high proportion of fertile tissue, and the sterile tissue is very small, whereas the members of class Bryopsida, Funaria, Polytrichum, Pongatum have the most complex sporophyte with a very high degree of sterility is there. The total evolution of sporophyte in bryophytes as such is considered to have taken place progressive sterilization of the fertile tissues. Here two theories are very important in this sporophytic generation. The theory of regressive evolution. This theory is known as regressive or retrogressive theory. It was supported by the several scientists like Church in 1919, Kashyap in 1919, Diobel in 1930 and Evans in 1939. According to this theory, the simplest sporophyte of rhexia compressed a simple capsule is the most advanced type of with as evolved by the simplification or progressive reduction of the complex of sporophyte masses. The example Funaria, Pogatum and Polytrichum. The theory of regressive evolution, rhexia is not a primitive one, but it is a more advanced type. It is simple capsule is believed to have evolved by descending the or regressive evolution. These progressive simplification, they think that the primitive sporophyte is like that of mosses, for example Funaria, from which the sporophyte of Marcantiales, Gingermaniales and Anthoceratales have been evolved by reduction of the tissues. The scientist Church in 1919 suggested that the hypothetical ancestor of bryophytes had foliase sporophytes, which complex assimilatory tissues and an epidermis which functional stomata. From such a plant evolution progressed in the recessively as. This one is the stage progressive, the semi-parasitic foliage sporophyte gradually lost its leaves and become embedded within the gametophytic stage. Gradually, reduction of photosynthetic tissue in the sporophyte. This restriction or absence of stomata, that is called example liverwort, only in the apophysis region, Funaria, Polytrichum. In sphagnum, the stomata of apophysis are non-functional and become rudimentary. Here the capsule of most of mosses, hornworms and some of the gingermanials are multi-layered which subsequently become a single layer, Markantia, Rixia, etc. These are the progressive reductions are there. Along with these changes, there was gradually elimination of the seta and foot. All these changes are accompanied by the progressive increase in the fertility of the sporogenous cells. Evidence from comparative morphology and experimental genetics support the view, and the simplest sporophyte of Rixia is not an advanced but it reduced structure. These are the both stages are very important for the sporophytic tissues, and in this one this also Rixia sporophytic primitive and but not advanced type of the stages. Coming to the these theories, approximately total 50% of sporogenous cells from spores and the remaining 50% of the sterile elaters, all characteristic spirals or thickenings are there, and sometimes bulbous foot, elongated sheet, a single layered capsule wall, and large number of elaters are formed into the sterile constituent of the sporophyte. The result of this complexity, the mechanism spore dispersal has been established, and the further sterilization of sporophyte tissue can be seen in the other classes of the bryophytes also. But jacket and capsule cells also differentiated in the fixed sterile cells are present. Here the elaters, these elaters may be reduced to be comparatively small and present in the total sporophytic tissues. The total mature sporophyte of the Anthoceras and continuous of the bulbous foot, horns or bristle like capsules are present. The capsule grows continuously due to the activity of the intercalary meristems. These Anthoceras, the main important of the intercalary meristems are present. That is where the entire endothecium from the sterile columella. The amphithecium forms the capsule wall sporogenous cells. The total wall capsule is multi-layered and the surrounded by the band of the sporogenous tissue is present. Here total increasing sterilization of sporogenous tissue, simple and sporophytic tissues are there in the Rixia. This is most complex is present in the Funaria. These can be arranged throughout the different stages. First stage, the first is a simple stage, that is a sporophyte among the bryophyte, example Rixia. In all species of the Rixia, the sporophyte always consists of the very simple capsules being to this one seat and futa is present. All the second stage example for the Courcinea belonging to the hepatic apsida and sterile foot also develop. These are the single layer, but inside the capsule sporogenous cells instead of the spores are nutritive cells are there. And next one is the further sterilization is the sporocarpus, where the sporophyte as a sterile bulbous foot are present in this one. Here the sporogenous tissues are the two theories are most important. This is known as regressive evolution. In this theory, progressive simplifications are there. This theory was proposed the scientist Kashyap year 1919, supported the church in also 1919, and Giobel 1930, Evers 1939. These people to explanation of different species, different types of sporophytes. Rixia sporophyte primitive, but it is not primitive, but it is no more advanced type, and the simple capsules are there believed to have evolved by descending, and regressive evolutions are present. This is called the progressive simplification. They think that the primitive sporophyte is like that of mosses. The example Funaria, from which is the sporophyte of Marcantiales. Gingermaniales and Anthoceratales have been evolved by the reduction tissues. The Church 1919 suggested that the hypothetical ancestor of Bryophytes had foliage sporophyte, which complex assimilatory tissues. This is the epidermis, which functional stomatas, all are the evolution progress regarding by this the theory of the regressive. Here many stages are there. There is a semi-parasitic stage, gradually reduction of the photosynthetic and restriction of the absence of stomata. Next one is the apophysis region, and next is the capsule of most mosses. These are all the stages, different stages. Along with these changes, there was gradually elimination of the seed and the foot. All these are changes accompanied by the progressive increases. These evidence from comparative morphologically experience and genetic support of the view and simplest sporophytic of the Rixia. It is not advanced, but it is the reduced structures are present in this sporophytic stage. These are theories also involved in the alternation generation of bryophytic sporophyte. Although these the dominant is the always gametophyte, but that sporophytic also very alternation generations are present in the bryophytes. Thank you.